All right, so what you want to do is take that image of this paramecium and clibinomonas and look at those functions of life that you see on 9 and 10, and just write on the diagram what you see, evidence, and it's all guided out there for you. But you can see lots of different things. You see this food vacuole, actually it's called the oral groove right here. Ooh, pen is messing up a little bit, and that's where it gets its food. Whatever kind of like a, the food. Nice little hamburger right here. You can probably tell. Vegan burger, of course. Food goes in there, and of course that's nutrition. Living things need to gather food somehow, either photosynthesize or eat things. Nucleus, be able to reproduce, pass on information, DNA or RNA. Let's see, metabolism. You can't really see the mitochondria, but all living things need to be able to break down their food into ATP, usable form of energy. Pretty cool. You can see all the little cilia all over here. Yeah, the cilia. Here you can see them. And that's to help it move around, so response to the environment. You see that even in plants. People don't think plants move, but plants can slightly move to respond to light and grow their roots to find water, and their seeds move around. Anyway, homeostasis, there's a pump called a contractile vacuole that pumps water in and out. Make sure it doesn't burst, and make sure it gets enough water. And with the clibinomonas here, pretty cool. You can see, almost looks like a little mustache. But you see a couple flagella. Of course, that's for response and movement. Here you can make out the outline of a nucleus, reproduction, pass on genetic information, and it's green. So you can see it photosynthesizes. So stem cells. You haven't heard about it in a while, but it comes up every once in a while. People get really worked up over embryos because the very beginning of a life form, a human life. So what we do with them, we can do really good things research-wise, but then some people think that's ethically incorrect. So, it's very important, and IAB really teaches you to look at the ethics of all the issues that we face and think about what's really happening, and look at all sides of an argument, because usually there's not either a black or white answer. There's a lot of gray area. So I want you to really focus on the gray. That's the hard part in your life. Black and white doesn't happen that often, but gray happens a lot. So this chart, kind of gives us a good idea of that. So make sure you fill this out on yours. But embryonic stem cells have some advantages. There's that controversy, so they cause some difficulty with researchers using them. But they have the best growth potential, and they can become anything. They're much easier to make into whatever a researcher wants to, but they're more likely to turn into cancer, to tumors. And that's already happened on a couple of trial runs. And they've been around, so you don't want to do a treatment, use stem cell therapy and then end up making somebody worse. That's not what we want to do in medicine and treatment. Since they've been around long, you have less mutations accumulated, which anything in an adult or even for an infant, you could have mutations accumulate. If we take a stem cell from just a regular person and then we implant it in somebody else, get a treatment, they could be genetically different, and that can pose a problem for rejection. And normally you have to kill the embryo to use it. You can do these things where you take a couple cells out and the embryo survives. So that's something that researchers can do to avoid that ethical dilemma. But just the idea of having embryos separate out there waiting in a freezer I would argue a lot of people would say that's not quite ethical. And that would bring in a whole bunch of issues with, say, in vitro fertilization, when a couple, donates their sperm and egg to have a child when they can't do through regular fashion. Is that ethical? That used to be highly questioned and thought to be wrong, but now it's pretty well accepted. So many people do it. You can also get stem cells from cord blood, and easy to get, easy to store. There's already a market for it. If you use it for yourself later on in the road, no way you're going to reject it. But they're limited in what you can do with those cells. All right. And hey, it's going to get thrown out anyway. Might as well use it, right? And then adult stem cells. These are things like right now, either of us, we could burrow down into our bones, into our marrow, and get some stem cells. But it's expensive. It's tough to do. They're not as differentiatable, different tissue types, but less chance of having tumors because of that, and no rejection problems. And I can't remember what this is. But anyway, those are the basic, you may have heard about stem cells before and wondered, what's going on with them. And this is a good, description of the different types of stem cells and their issues. So you should be able to draw a diagram of a microscope I have here. So what you look through is the ocular lens. Let's see if I draw a little eyeball here. So you look through here, and these down here are objective lenses. There's 3 of them, low, medium, and high. They'll say 4, 10, and 40. Remember, this is 10. So lowest power is 40, then 100, 400. Pretty good. We can see some pretty amazing things. 400 power magnification. You have the stage where your specimen is, and underneath you have the diaphragm. And very overlooked part of the microscope is the diaphragm. You can fine tune the amount of light comes through, and it really helps with being able to see your specimen. You got your coarse adjustment knob, using it under the low power. You use coarse, and then when you get up to medium and high, you want to use the fine focus, which is down low. And don't forget your light source. Make sure that's on, or you won't see anything. Oh, okay. I just skipped that. Sorry. I'll go back and add to that in a second if I have to. But this is how you do, I think it's how you focus it. So go through those steps. Another thing I've been doing, especially today, we did banana cells, and we finished up the red onion cells. Only draw a couple cells. Don't try and draw everything. Don't draw a circle and then stuff inside of it. Just don't draw that circle. Just draw the stuff inside of it. Because if you draw the circle, you're going to start to try to fill it up with everything. Nature hates a vacuum. And just draw your cells. Okay? Look back and forth, back and forth, back and forth to really see what you're drawing. You can take a picture with your phone. That can make it easier. And I want you to label everything. Normally, the cell wall, the membrane, the cytoplasm, and usually the nucleus are all visible. Of course, the cytoplasm. So label those. Okay, get used to labeling. This we did today. You can calculate the magnification of a scale drawing here. Like, here's the scale. It gives you a length, and it gives you the actual length. So what you do, remember, it's measure over the actual. Take your ruler. This is my wife's ruler. Her name is Danielle. So what I did is I measured this on my screen, and it measured 18 millimeters. And guess what? I know it's 100 microns. So I just converted the 18 millimeters to microns, and then cancel out the 0. It's 180 time magnification. Okay, remember we talked about here. If we're going big to small, we're going to move to the right. So that's what we did here. Big to small. Big meaning the millimeter. Small meaning micron. 1, 2, 3. That makes 18,000. And just millimeter, make sure you remember, is 10 to the third. Microns 10 to the 6th. Remember $1 million? 6 zeros. $1,000? 3 zeros. Okay? try to use that. Check out, pretty cool. That's from the last video, I think. And here we have red blood cell, very, very cool. And what happened here? Oh, so same thing. I calculated the magnification by measuring what I saw here. I measured the distance, which was 14 millimeters. And on your screen it can be different. It doesn't matter as long as, on the test, the IB exam, you'll just measure whatever it gives you and divide it by 2 microns, convert it to millimeters. 14,000 divided by 2 is 7,000. And check it out. What I did, you'll see in a little bit, I'm going to have you write down what's the length. So what I do is I did a proportion for that. So I put 14 over 2, and then so that's the measure over the actual. So then I put the measured, I measured here to here, and for me on my screen I measured 37 millimeters. Remember, this is millimeters, this is microns down here. And I basically just cross multiplied 2 to solve for x down here. So 2 times 37, then divide by 14 gives me 5.3. So that's the actual. If you look at that, it makes sense. Look, 2 is about this, right? So it's at least 4, like and a half. So 5.3, that seems pretty reasonable. Always check your math, see if it seems reasonable. And that's what we had. You can look back on those slides. I think there was 7,000x with a magnification. The aseptate fungi was 180x, and it was the red blood cells 5.3 microns wide. And here's just some cool photos. This is Robert Hooke, very famous. Hooke's microscope. Beautiful. Look at that inlay. Oh, nice. And then, I don't know how, I think the lens is here. And this is some of his first drawings. This happens to be core. And this is Anton van Leeuwenhoek. He had a different design where he had the lens right here. And on this little needle point, you would put the specimen to move it around into focus.